Hello, this is Sarfaraz Niazi, bringing to you a topic that is close to my heart, but it is controversial. Over years, I have come to conclude that it is not necessary to test a biosimilar candidate in patients to prove its safety and efficacy. And this time, I'm using a very different approach to convince you, a Socratic approach, a questioning approach, until we are all satisfied. So, Socrates brought his pyramid long before FDA thought of it. His questioning and his modus operandi is very simple. No matter how obvious, you must always ask a question: why? Why am I doing it? Why am I doing it? And you reach a point where you can either have no more questions or no more answers. The case closes. But he also taught us something very important, okay, which I want you to remember as you deal with regulatory agencies. If you don't agree with an agency, you ask a question. I found FDA and EMA to be extremely responsive, extremely concerning when you ask a scientific question. Why? Why should I do clinical efficacy testing? Why should I do clinical immunogenicity? What is the residual uncertainty? Sometimes we do, sometimes we don't. But that's what Socrates teaches us to do. So let's say today, if Socrates were to be introduced to biosimilars, he will first ask, why you call them biosimilars? And I'll tell him, these are biologic products that happen to be similar. Case closed. It is not a similar biologic, which totally changes the definition. Why do we need a reference product? Because I don't have a standard to match with. It's a moving target. And my job is to make it just as bad as the standard, which is another reference product. You'll be satisfied. Why do you have to do such extensive analytical similarity assessment? Again, I'll tell him this is a non-fixed structure, it's a moving target, and I'm trying to match a moving target against another moving target. So I need to have many views, many visions, many scopes. He will say, okay, that's fine. I think that makes a lot of sense. Why do we do clinical pharmacology testing in humans? And I said, that's important because I'm not using it to prove anything other than that the two products have a similar molecular structure. It's how body sees my molecule and how my molecule sees the body. Can there be a better test for that? Probably not. And this testing is based on my very simple question. Today, Perseverance is sitting on Mars and telling me there's a life or not. Okay. And we are sitting on Earth disputing whether we can tell if the two molecules in two different test tubes are same or not. He would not agree. He said you should be able to do it. So what's the problem? The problem is us. So I got this 4 things to share with you. So if you are able to look at the bottom left, know that this is a picture, not a video. Something is moving. On top of left, both orange circles are same size. The X you see there is only one color of red in the screen. And on the bottom right, on every circle you see has same color. I'm showing it to you only to bring a reason why we need to use orthogonal testing. And that's the topic that I want to explore with you, is that we have a limited vision. And it is not only our eye vision, it's the instrumental vision also. So let's take a look at how this vision has complicated the development cycle and brought a boatload of confusion among the developers. So I looked at all the biosimilars that have been licensed by FDA. And in summary, I found there was no consistency in how many studies are submitted for the same molecule. There are 4 adalimumabs approved, and one of them had 31 studies, other had 105. Trastuzumab, there were again 5 of them, and there were anywhere from 48 to 111 studies. And the 4 infliximabs, have 48 to 80. So my question is, if there was a consensus on how to establish similarity, then there will be generally a similar number of studies required. If somebody is doing too little, absolutely not. FDA will not accept if there's not enough studies. If somebody is doing too much, I'll give you an example. 27 animal pharmacology and toxicology studies that were submitted to FDA, FDA refused to review them. One company, very large name, submitted 9 animal toxicology studies, more than what is required for a new molecule. And we said, no, thank you. This is the background. We still carry a mentality of developing a new molecule. It's not. We are supposed to show similarity and not characterize the effect of it, because this is where the difference comes. So my first question, which I concluded after looking at all the reports, was that if the analytical assessment was acceptable, no animal toxicology study failed. And if the no clinical efficacy study failed if the product had first met clinical pharmacology. In fact, FDA wouldn't even allow you to go to clinical efficacy unless you have proven clinical pharmacology. So on a very simple argument, philosophic argument, if a study does not fail, it means 2 things. Very simple, that because the products are same. Why are you testing them? Or the study is not sensitive enough to tell the difference. Why are we testing them? And that's the basis of my argument today, that we don't necessarily need clinical efficacy to prove similarity. First of all, I have a serious issue with using animal toxicology in development of biosimilars. Rodents are used initially in the development. Yes, a rodent and us share some genes. Actually, we share a lot of genes, to be frank with you. But the point is, they don't have the receptors, correct? There's no way to tell the dose to give them. I get very annoyed when I see agencies telling you, well, give 5 times the human dose. Or if you can't do it in monkeys, just do it in rodents. Okay. I think we have to understand that we are not trying to establish safety by dosing. We are also recommended that we study the toxicology at the higher end of the linear dose. None of the study failed. It could not possibly fail. Okay. I think testing in rodents for toxicology is a pure waste. And I don't want to go into ethics parts of it. That's another subject for some other day. The best you can do and you should do is get a pharmacology done in animals, larger species. Monkey will be the best one because it also has some receptors that can be responsive. But the purpose here is not to look at the safety, but look at how a body sees the molecule. It is a part of analytical similarity testing. FDA requires such testing for MABs, but I am suggesting that we should do this for all products and not leave the little guys on the left alone, okay, because it doesn't add any value to our testing. And now I come to the topic which I think we are talking about today, and that is, should we do the testing in human patients? So I don't want to go into too many technicalities and jargon, but M1 is the response which we know because we have a reference product approved, which was approved against a placebo. And it was a phase 3 study. By the way, I very often I tell in my talks, there is no phase 3 in biosimilars. There's no phase 1 in biosimilars. But every company, big or large or small, keep telling me, oh, I did a phase 3 study, phase 1 study. No, phase 1 study has nothing to do with clinical pharmacology study. Okay, but anyway, that's more on the semantic side, but maybe something to talk about how you think of it. M2 is how much it can vary. You cannot test something without first establishing your M2. How much is 10% variation okay? 20% okay? 50% okay? 100% okay? I asked every agency. Nobody had an answer. The only answer I got, it's based on a clinical judgment. So, fine. In one jurisdiction for the same product, the jurisdiction was 20%, and in other it was 48%. And these are the real numbers. What do you say that? There's no way possible for us to determine on an arbitrary basis because we never have studies which were based on multiple dosing of the different dosing, not multiple, different dosing of the reference product to see what is the variability involved with dosing. So now you're telling me that you're also going to use a single study among 4 or 5, possibly more indications, only one study, and only one M2, which is based on my clinical judgment. And then you're claiming that this clinical judgment will apply to all indications, regardless of the M1 value. It doesn't make sense. One study will extrapolate if you're testing it. But here's where we get into some really rough grounds. Okay. Number one is, let's say you're developing an anti-cancer MAB. You're never going to get a naive patient for cancer treatment. A good number of them will die before the study is over. The milestones, the markers sometimes are not there. And it takes you years to complete the cycle. But it's still, if you know just even basic statistics of it, if you're trying to differentiate a product against placebo, it is so much easier than trying to track down 2 moving targets. The alpha risk is very high. Alpha is what I call FD risk is high. Okay. How many subjects you need to prove it? In reality, if you do a phase 3 type study to compare the 2 products which are variable, you may end up needing more subjects than were required to even establish the first proof of efficacy. So my argument is simple, okay? The purpose of biosimilars was to reduce the cost and time to get into market. On top of it, now we have an issue which is to do with non-inferiority. Is it okay for a product to be superior in clinical response? Answer is absolutely not, because with a higher efficacy comes a higher toxicity also. My argument is very simple. If you can find a clinical protocol reasonable that will take out any uncertainty, so you have to go back to FDA and ask them, where is the uncertainty? Now, here's what happens, okay? If you don't ask, then of course, you end up doing these studies in all likelihood for perhaps promoting your product or presenting it to clinicians. But before I close my talk, okay, I want to tell you that the idea here is not a stepwise approach to approval, but a stepwise approach to rejection of biosimilars. If it doesn't meet analytical similarity, reject it. If it doesn't match pharmacology in animals, let's say 5 or 6 monkeys, simple PK, reject it. If it doesn't match human pharmacology, it's rejected. So we must first ask, okay, do we really need to do this study? In my analysis, I found that the developers were just too eager to present to FDA or EMA. I don't want to give numbers because sometimes you can identify who the company is. But if I'll take a hypothetical example, you're developing a cytokine and you go to FDA and say, I'm going to do 10,000 patient study. So there's an official response you will get from EMA and FDA is called hallelujah. Don't do this. There's no need for it. But before I close my talk, I want to share with you something very interesting that has to do with my recent work with FDA in creating a very novel clinical pharmacology protocol. A single study will give you all the answers. My testimony that was done by Dr. Sarah Yim, she has been very kind to me and always listened to my rhetoric. I say rhetoric, okay. So you can significantly improve your analytical similarity testing through pharmacology, clinical pharmacology. You know very well what FDA has said enough time. They prefer in silico approach. And the one point which I want to share with you is that the purpose of this study is to compare, not characterize. Do not select a population which is diversified. You don't need to. You can narrow down the population significantly and be able to get the product tested. You can also use the two-tier system where you can do a small number of subjects and then more and add them together. I think both FDA and EMA will allow you to do that to improve your study power. And in the end, I only want to tell you something that if you make your arguments rational, people will believe you. If you think in your mind that the only way to tell the biosimilars are safe and effective through putting it into patients, then why bother with all the other testing? The goal is not to do clinical efficacy testing, because I think it will be on the borderline unethical, impractical, irresolvable, and it does not add any value to the safety and efficacy of biosimilars. I want to thank you all for listening to me. I'm sure many of you would not agree with me, and I think that's exactly where Socrates comes. Okay, you ask question why and why. I've come to conclude this is the best way we can promote biosimilars. Again, thank you for listening to me. Have a nice day.